In this lecture, we are going to talk about part 3 of translation, and we focus on elongation. Main points of the lesson are matching tRNA anticodon with messenger RNA codon, peptide bond formation, and translocation of the ribosome. In the previous video, we discussed about prokaryotic and eukaryotic initiation. In this lecture, we focus on elongation. Elongation cycle adds successively amino acids. Elongation is essentially the same process in prokaryotes and eukaryotes. When the entire ribosome is assembled around the initiator tRNA, messenger RNA, and the second charged tRNA can be brought to the ribosome and bind to the empty site, which is called A site. This requires an elongation factor that is called EF, and EF binds to the charged tRNA and to the GTP. Elongation factor is responsible for catalyzing the binding of an Aminoacyl-tRNA to the ribosome at the A site. What is Aminoacyl-tRNA? Aminoacyl-tRNA is tRNA which is associated with an amino acid, and Aminoacyl-tRNA is charged tRNA along with particular elongation factors deliver the amino acid to the ribosome for incorporation into polypeptide chain, and this polypeptide chain is produced during the process of translation. Elongation cycle that takes place in 3 series of events. Number 1 is matching tRNA anticodon with messenger RNA codon. Second is peptide bond formation, and number 3 is translocation of the ribosome. First step is matching tRNA anticodon with messenger RNA codon. This is charged tRNA which is present here in the A site, and the sequence of anticodon is UGG, and this sequence is matched with the sequence of codon in messenger RNA, that is ACC. So, this is step number 1, that the complementary nucleotides in anticodon and codon, they are matched. This binding step requires several proteins, and these proteins are called elongation factors in addition to GTP. After binding the transfer RNA to the codon of messenger RNA, GTP is hydrolyzed, and elongation factor and GDP they dissociates from the ribosome, where it is recycled by another factor. So, this binding and hydrolysis of GTP is thought to increase the accuracy of translation. Step number 2 is peptide bond formation. It takes place with the help of peptidal transferase enzyme. What is the function of peptidal transferase? Peptidal transferase enzyme that is located in the large subunit of ribosome, and this enzyme catalyzes the formation of peptide bond between the amino group of amino acid in the A site and carboxyl group of growing polypeptide chain in the P site. So, this enzyme breaks the bond between the growing polypeptide chain and the tRNA at the P site, and now this tRNA is empty, no longer charged, and peptidal transferase it forms a peptide bond between the amino group of the amino acid in the A site and the carboxyl group of growing polypeptide chain in the P site. The overall result of this is to transfer the growing polypeptide chain from P site to A site with the formation of peptide bond. Energy cost for peptide bond formation is 0 GTP, and this reaction is spontaneous. It does not require energy because ATP transferred energy during the formation of Aminoacyl-tRNA. So, the peptide bond formation only requires one enzyme, and that enzyme is called peptidal transferase enzyme. This enzyme is not a protein, but a ribozyme that is RNA component of large ribosomal subunit, that is called catalytic RNA. Step number 3 is translocation of the ribosome. After the peptide bond formation, ribosome it moves relative to messenger RNA and the tRNA. So, ribosome usually moves in the direction of 5 prime to 3 prime from start codon to stop codon. So, during translocation, next codon in messenger RNA it shifts into the A site, and tRNA with growing polypeptide chain that moves from A site to P site, and the uncharged tRNA from P site it moves to the E site, that is called exit site. It will be ejected in the next cycle. This elongation process continues with the new amino acid added here in the A site. Then a peptide bond formation takes place between the amino acid and the growing polypeptide chain in the P site. So, the polypeptide chain is shifted from P site to A site. Again ribosome it moves to the next part. So, elongation cycle continues with the new amino acid added here in the A site. So, tRNA moves through the ribosome in the opposite direction, because ribosome is moving in the direction of 5 prime to 3 prime, and the tRNA in the ribosome they are moving in the opposite direction. So, the tRNA in the A site it moves to the P site, and from P site finally moves to the E site. From there it exits the ribosome and joins the cytosolic pool of tRNA, which can be charged with another amino acid and then used again. Let me briefly restate the main points. First step of elongation is matching tRNA anticodon that binds with messenger RNA codon. So, the appropriate Aminoacyl-tRNA recognizes the codon in the A site and binds there by base pairing of its anticodon with complementary messenger RNA codon. Second step is peptide bond formation, and peptide bond formation that requires one enzyme that is called peptidal transferase. This enzyme is located in large ribosomal subunit, and that catalyzes the formation of peptide bond between amino group of amino acid in the A site and carboxyl group of growing polypeptide chain in the P site. Third step is translocation of the ribosome. So, after the peptide bond formation, ribosome moves relative to messenger RNA and tRNA. So, translocation involves the movement of ribosomes from 5 prime to 3 prime direction along the mRNA molecule.